The following was recorded in front of a live studio audience at the Studio 21 Podcast Cafe. This is the United Podcast Network.
This is Voices in Validation, brought to you by IVT Network. IVT Network is your expert source for life science regulatory knowledge. Voices in Validation brings you the best in validation and compliance topics. We interview industry experts from pharma, biotech, med devices, and laboratories. Here is the host of Voices in Validation, Stacey.
Thank you, and welcome to another episode of Voices in Validation, the podcast bringing you timely interviews with industry experts from pharma, biotech, medical devices, and life science manufacturing. Each week, we discuss trending topics and industry best practices, and this week, we are pleased to have with us Dr. Tim Sandle. He's the head of microbiology and sterility assurance at Bioproducts Laboratory Limited in England. And he is joining us today to give us his thoughts and some updates on the European GMP Annex 1, talking about what the final draft actually reveals. So the long-awaited revision to the European Annex 1 draft documents has caused much anticipation, Tim. So I am very interested in this conversation, as are most of our listeners. So thank you so much for joining us.
You're welcome.
So across regulated industries, this is something that people who are doing business in Europe have been waiting for and very interested in these draft documents. They haven't been approved yet. There is a 3-month period of time, I understand, where people can make comments. So this podcast is very timely and hopefully will shed some light on a few things for our listeners so that they do have an opportunity to respond if appropriate. So let's kick it off for the benefit of our listeners with an update or a little bit of history and background on what Annex 1 is and why it's so important in pharmaceutical manufacturing.
Okay, that's a good question, Stacey. Thanks. Annex 1 has a long history. It first started out as something called the Orange Guide that was linked to UK GMP and first issued in 1971. And since then, it's grown substantially, became part of European Union GMP in the 1990s. And what it essentially does, it covers the minimum regulatory expectations for the manufacturing, control, release and distribution of sterile product manufacturing. So what that embraces is terminally sterilized products, aseptically filled products, and also all the variants such as blow fill seal technology. And the reason it's important is that it applies to anybody who manufactures a sterile drug product within the European Union, but also for anyone who wants to distribute a sterile medicine into the European Union. The other thing that's important, particularly for people in the US, is that it was developed, this latest revision, through the PICS process, the Pharmaceutical Inspection and Cooperation Scheme. So US FDA were part of the review process. So it's probable that a lot of what's in Annex 1 may well end up in the eventual revision to the 2004 FDA aseptic filling guidance whenever that comes up for review. So although it's European and it has a big impact, it could well be the basis for a true global sterile products manufacturing standard.
Right. Yes, I agree. I think that as it stands right now with Annex 1, people are paying attention globally because many companies do distribute, to your point, in the UK, in Europe, whether or not they're physically located or producing the product there. But it seems like global regulatory agencies do talk to each other, and oftentimes when they come up with regulations that make sense for the industry, it's adopted across the board in some form. So you mentioned that it started as the orange guide. I didn't even realize that because the original draft guidance of the Annex 1, GMP Annex 1 as it stands now, I believe was released in 2008. And you covered the draft revisions, how they've evolved over time. So I know you've spent some time reviewing the current documentation that's out there for review. What are some of the most pertinent and impactful changes from the previous version, which I believe was released in 2017?
Yes, and even a bit before that. So this is the first major update for 12 years. There was a very small update in 2009 where a grade A air expectation for oversealing vials was added, but essentially this is the first big one. And we got wind of this in 2014 when a concept paper was issued. Then there was a 3-year wait and the December 2017 draft appeared. And the reason there's been now another 2-year delay was because of the level of industry comment. I think the December 2017 draft received something like 6,200 comments. So obviously that took time for the European Medicines Agency to shift through. So what we've ended up now is with a new draft that's actually a couple of pages bigger, and the kind of the key sort of standout things are kind of grouped around 4 kind of key factors. The first one is the embedding and criticality of the concept of quality risk management. And here the Annex is connecting with ICH Q9 in particular, which came out, I think, in 2005. It also attempts to show how sterile manufacturing needs to link very strongly into the pharmaceutical quality system. So there's a lot in there about documentation, validation, traceability. The third kind of key point is this concept of a contamination control strategy, which I think we'll probably talk about later, but that needs to flow through every aspect that's done. And the final point is that although Annex 1 pertains to sterile products, there's actually a recognition that there are elements within Annex 1 that non-sterile manufacturers might well want to have a look at. It's not saying that non-sterile manufacturers have to abide by it or they're going to be regulated against it, but there are some general good practice guidance that might well be applicable to the entire pharmaceutical industry. So just as a broad overview, it's risk management, pharmaceutical quality system, and contamination control strategy that really stand out as the key features.
Thank you. So it seems like it is more far-reaching than some people might have thought. I hear a lot of people talking about it, whether they've read the actual documents, I don't know, as a contamination control or sterilization issue or guidance. But it sounds like it's much more far-reaching than that.
It is broader than that, and for a sterile manufacturer, it would cut across almost every kind of GXP role, whether someone's just involved with distribution or warehousing or packaging. It still embraces every part from the purchase of a raw material to the distribution of a finished medicine to another part of the world.
Excellent. So you mentioned, obviously, we are going to talk about the contamination control piece because I do think that is a big part of it. And with that, I wanted to talk about clean rooms and clean air systems and get your thoughts on why now, like why this draft document or this draft guidance now, as opposed to 10, 20 years ago. What was lacking, in your opinion, from previous versions in regards to contamination control?
I think it was just an all-round lack of detail. And it's also an acknowledgement of the advancement in sterile manufacturing technology. So 12, 13 years ago, there were isolators, but they weren't as widely used. Now there's an attempt to nudge industry towards barrier technologies. I think there are a lot of regulators who used to go into facilities and find poor environmental monitoring. There was a lack of proactive risk assessments. So there's and there's also rapid microbiological methods come on the scene. So there's a lot of developments that have taken place, but also, without getting too deep into European Union politics, there's a lot of different member states that have particular views. There's lots of stakeholders. So just producing a document like this is in sort of at least a 5-year cycle. And that's just to produce a draft, which then goes for comment, and then there's the next version. So, it's a convoluted process, is the time, and also just acknowledging that there's been shifts in thinking and there's been the advent of new technologies.
Yes, of course. I mean, that is a big factor in itself, just all the new technologies that have come about since the original version in 2008. And we certainly can see that a lot of detail must have been added to this revision, because it went from something like a 16-page document to more than 50-page document. So certainly expanded in
definitely
what's included.
And rewritten as well. So it's not just adding to, virtually very little of the text is the same.
Right. Okay. New definitions and a lot of new things happening. So I've started to read some of the comments, and there are things that people have concerns over, or that are maybe just sticking points in the continued distinction of some terms and terminology, verbiage, that kind of thing. And I think you yourself had pointed out that the distinction between viable counts and non-viable particle counts in this latest draft as a conventional particle count, it brings some concern to you, as conventional particle counters don't determine whether particles are viable or not viable. They're just counting particles. Is that correct?
That's correct. Yeah. So the Annex makes reference to viable particles, which is in itself a slightly odd term, because there may be particles that are hosting viable microorganisms, but there aren't many free-floating microorganisms in themselves, and microorganisms on surfaces are microorganisms, not necessarily particles. That's the first issue. Second issue is that it does talk about non-viable particle counting, and that's alluding to the conventional optical particle counter. But you're right, that those counters detect a number of particles in a given volume of air. That's normally 1 cubic meter, and it will say how many there are, and it will be able to break down the sizes of those particles. But it has no way to say whether those are viable or non-viable particles. That's the first thing. And also, although the Annex does refer to rapid methods, what it's not doing here is acknowledging that there are rapid microbiological methods now established on the market, which do have the ability to differentiate between what are potentially particles that are holding viable organisms, and then what are referred to as inert particles, as in particles that are not hosting viable microorganisms. And these work on spectrophotometric technology, and they will one day have a key role in being assessing grade A environments in particular. So I think there's some terminology issues and a little bit more development with thinking that can also go into it. And there's also a little bit in terms of although the Annex is acknowledging that environmental control is important, there's still a little bit over-reliance on environmental monitoring without, I don't think, properly recognising some of the limits in terms of small sample sizes and the issues with the accuracy of some of these instruments.
So if the draft passes with the verbiage that it currently has, and they don't clarify that distinction between viable and non-viable, and I did hear you say that there are advanced technologies out there that can, counters that can now detect between the two. I'm thinking probably those are costly, and in the short term, I'm wondering what do you think that means for pharmaceutical companies? How are they going to get around that that verbiage, or produce the correct data given that verbiage? And what do you expect regulators will be looking for at that point?
I think this is probably one aspect where it's a little bit more carrying on as things have been. So there be conventional particle counters run close to conventional environmental monitoring samples, and there won't really be a clear relationship between the two. Obviously, the big difference between current US standards and European standards is with the sizes of particles that are looked for. So the FDA guidance is for screening for particles that are equal to or greater than 0.5 micron, whereas EU GMP requires that, but also has this additional requirement for looking for particles of 5 microns and greater as well. So you kind of still got that difference in translation, but regulators will be looking for those sort of concepts and that kind of level of monitoring, but it's very, very difficult to kind of correlate the two together.
Okay, thank you for that clarification. Another spot that I noticed where wording might be of concern to folks is with barrier technologies. Can you provide some background on that and discuss the impact of the document changes on current processes in the lab? I know you've spent a lot of time in the lab.
Yeah, I mean, so the first thing is that the mention of barrier technology is important, and this is one of the paradigm shifts that's inherent in the new Annex, which is recognizing that environmental control is more important than simply monitoring, and the main originating area of contamination is from people. So by constructing barriers between people and grade A environments, excluding personnel from grade A through the use of barrier technology is a very important concept. But it could be argued that the Annex doesn't go far enough. It's a slight nudge towards barrier technology. And then with barrier technology, there's the distinction between isolators and restricted access barrier systems, or RABs. And with RABs, there are many very good RAB systems out there in terms of closed RABs devices. But there's also a number of open RABs devices where there's still the requirement to do grade A open door interventions. And it really needs to be emphasized that there's no such thing as a low risk or safe intervention. Every intervention does potentially put product at risk. So there really needs to be, I think, greater steer towards closed RABs or isolators. Although there is a slight enhancement with all filling equipment, where the Annex is now asking that after the setup process, and you could argue that the setup of a filling line is the biggest risk because it has the highest level of personnel involvement. But the Annex is now saying for the setup process that there should be sporicidal disinfection undertaken immediately following the completion of setup.
So just in follow-up to that a little bit, you talked a couple times during this conversation about environmental monitoring. And then you just mentioned personnel requirements in connection with barrier technologies. And I'm wondering if we are seeing that, where else are we seeing that in these draft documents where they're talking about personnel requirements and sort of the impact or the connection to environmental monitoring? Because it seems like that would be a big issue, right? We see a lot of human things happen as a result of human error.
Yeah, so obviously there's a key emphasis about really saying that people have no place in a grade A environment, but the personnel section of the annex has been strengthened quite a bit, and I think it's potentially the largest section. It only runs on to 3 or 4 pages about personnel. So some of the areas where personnel factors have been enhanced include, there's quite a big paragraph saying that people who work in pharmaceutical facilities now need to demonstrate that they have the knowledge and skills required to do the job, and that includes an understanding of basic microbiology. Even if someone's job is to just move things from one place to another, they need to be conscious that they're doing things in terms of slow, deliberate movements and avoiding cross-contamination and so on. Another area is with gowning, and there's quite a lot of discussion about the process of putting on a clean room suit needs to be done in a very controlled manner. So with the gowning test, there's an emphasis on that every year people undertake a test, and there's microbiological samples taken. It's maybe contact plates of the suit and finger dabs, and also there's a visual element where an independent assessor is watching that the leg of the gown doesn't go on the floor and so on. There's also a lot of emphasis about people working in aseptic processing areas being fully enclosed. That means no exposed skin whatsoever. So that's a shift away from some places where they may have slipped on a pair of safety spectacles, but now it needs to be fully enclosed goggles. There's a lot of conversation around gown integrity checks as well. There's also a strengthening of how people get into changing rooms. So there's the requirement that no outdoor clothing enters a grade C or grade B changing room. So this requires a changing room further down line where people would need to put on some kind of clean room scrubs. And the annex even goes so far to say that at this point, clean room socks should be worn. So people's everyday socks don't end up going into the clean room. And there's also then a requirement to assess the suitability of the gowns. So gowns should be assessed that they're containing any shedding. So gowns made of the right material and the surrounding temperature isn't too high, that people might be perspiring and shedding, and also that the gowns are not shedding fibres either. So there's just some examples of the controls around personnel. They're much more detailed, and this is one of the good things in the revised annex.
I mean, it is great to hear that they have paid so much attention to this particular area, because we do hear a lot about contamination coming from human error or, some of the things you're talking about. I'm wondering if, in follow-up to that, it sounds like there's going to be a lot of steps involved in people preparing to be in a clean room. And I know just from other, not necessarily lab, but from other parts of pharmaceutical validation and compliance, that when lists of to-dos are so big or processes are so long, shortcuts tend to get made. So what's your thought on that? Is this going to be feasible? If this passes as is this feasible for folks working in clean room and clean spaces, or do you feel like it's a lot to ask?
It's a lot to ask, but I think it is necessary. So just going back to the earlier point in terms of contamination, a number of studies, if you look at all the different studies on sources of clean room contamination, the level attributed to people is at least 70%. So, we can build very good working clean rooms, which will stay relatively clean and low particle levels. It's only when we put people in them that issues arise. So I think, if we are going to reduce contamination risks, stop recalls from occurring, we do need to control the people, but it will be more restrictive. So again, another thing in Annex 1 is reference to sort of validation, people and engineers and so on. That there's talk within the Annex about, particularly for aseptic processing areas, that only people who are qualified really should be going into those areas. So if there is a breakdown, then either you've embedded quality by design and have access panels that people don't need to go into the aseptic core so they can work on the equipment, or you might need to shut down for a day and then re-sanitize the facility, or you may need to go to the extra effort of having a couple of engineers or validation people already pre-qualified. And the qualification is quite extensive as well, because it's essentially inferring that you need to take part in a aseptic process simulation in order to gain the necessary qualification to work within an aseptic facility.
Sure. Yeah. And then that's something that people, that companies need to think about, obviously, as you mentioned, ahead of time, and then build into their process and just make it part of the protocol or their overall processes. I would think, because this is not something that you're going to be able to do last minute if it's such a time consuming thing for someone to become qualified in aseptic.
Certainly, yes.
Yeah. So,
and especially given let's say most companies only run media fills every 6 months, so that's easy to account for.
Right. So it's almost like having, obviously that's someone who's going to be wearing many hats, right? That's not going to be their only function with the company for sure.
That's right. Yeah.
I'm hearing also talk of pre-use, something new mention in the documents, I should say, of pre-use post-sterilization integrity testing. I understand from a couple of folks that there's maybe a more relaxed stance on this in the new documentation. What's your take on that?
Yeah, so I mean, the pre-use post-sterilization integrity test, or PUPSIT as it's generally called, it's been a long-standing controversial issue. So a few years ago, it was conventional to look at sterilizing grade filters before use and after use. But the PUPSIT concept is based on a theory of filter blinding, where immediately after sterilization, pores could open, cause a contamination risk, but then subsequently not be detectable at the end of use. But this is based on anecdotal evidence and unpublished data. So the first thing is, a lot of the industry have been saying, okay, this concept of PUPSIT may or may not exist, but can we see the data? And that's still yet to be forthcoming. And PUPSIT has presented a number of complexities for manufacturers in terms of buying new filter kits, and there are risks involved in how that is set up and how it's tested and so on. So there's quite a raft. And one of the biggest criticism from the 2017 draft, a lot of the comments among the sort of 6,000 odd comments were a lot of criticisms of PUPSIT. However, the Annex has offered it's almost like a two-tier approach now. So for large volume products, it's still stating that PUPSIT is necessary. However, for small volume products, it's saying that PUPSIT may now not be appropriate, and it offers an alternative risk-based approach involving things like assessing sterilization conditions, filter fragility, carrying out extensive suppliers of supplier audits of filter manufacturers and so on. So there is almost like a two-phase approach. What's kind of missing is any definition as to what is a small volume product. That's kind of a little bit left open to interpretation, but it does move it. It will satisfy an element, but I think there'll still be a number of critics have popped it out there, and I fully expect that to be part of the continuation of the consultation.
Tim, so you just mentioned more that they're taking a more risk-based approach, and based on our conversation so far, it sounds to me like a lot of the new draft text seems to really place a greater emphasis on quality by design, risk-based approach, which really the whole pharmaceutical industry is moving towards a more risk-based approach to manufacturing. It seems anyway. But it reminded me of the FDA's case for quality that they have yet to release, but have been working on draft documents for on the medical device side of things. Do you feel like this was intentional? Is it because the industry is moving more as a whole towards risk-based, or really that whole quality-driven process? There seems to be this whole quality approach that I'm seeing across the board in pharmaceutical and medical devices. And I'm just wondering what your thoughts are on that. As opposed to, it seems like for maybe the past several years, we've been more, or decades, we've been more reactive. And especially now that we're seeing more warning letters and findings coming out, people are trying to avoid those and move to. I'm just wondering what your thoughts are on that.
Yeah, so I think absolutely it's intentional, and quality by design is an important concept. So that's the philosophy of building quality at the conception or design stage, rather than trying to build in quality later on once a piece of equipment has been purchased. So it's thinking about, before I buy this piece of equipment, how might I validate it? How might I maintain it? What are the operational issues, be that equipment or a clean room or whatever. So I think that really is key, and there is a deliberate attempt to do that. And also, it also fits in with, makes it easier for a kind of a similar quality concept, which is the validation life cycle approach as well, because unless the quality by design has been there at the outset, continuously assessing something is going to become a lot more complicated as well. And also, I think it, again, I mentioned at the beginning, it also helps tie in the annex with the overall concept of the pharmaceutical quality system and about being proactive. So that kind of squares back with the talk about risk assessments as well, because one of the regulatory concerns is lack of risk assessment, but a second regulatory concern is that too many risk assessments are reactive. After something's gone wrong, it's let's try a risk assessment. But this is all about thinking up front. What are the risks? What might go wrong? If we buy this piece of equipment, it's doing things like FMEAs and embedding all of that down. So I think all that is clear from the annex. It's all about thinking before you actually act and do something.
Excellent. Yeah, and that makes sense, because as a company, setting yourself up for highest quality right from the beginning is certainly going to be easier than having to do an end run around and try to fix things, through CAPAs and just all kinds of change control going on, once the product's beyond a certain point. So I'm glad to see some of these things being addressed in the draft annex. So overall, because you had a chance to see the 2017 draft version as well. Do you see this 2019, 2020 draft version as an overall, an improvement overall to the 2017 draft, or what are your, in a comparison, what are your thoughts on that?
Yes, I think it's better. It's not perfect, but right. It is a big improvement. That some of the points raised from the consultation have been addressed. The language is less ambiguous, because in the earlier draft there were kind of weird things like it might say grade A or grade B, but you need to be much clearer whether it is grade A or grade B, because there's a big difference in the environmental standards between those areas. But there are a few areas of ambiguity where I think greater clarity would be needed. So around media fills, aseptic process simulations, there's still a lack of guidance about how big and how long they should be. There's a new requirement to add a microbiological assessment for clean rooms, which kind of makes sense for a new clean room, but could become problematic for recertification. I know there was one point raised from the 2017 draft about disinfectant residues, and the Annex not acknowledging that there's lots of low residue disinfectants out there, and unfortunately that's still in there. We've spoken about PUPSIT as well. But overall, it's a more solid document, and it's easier to read and easier to understand, and it's been grouped into more logical sections, and it's quite clear that it does, we've just spoken earlier about the importance of quality and risk, and that's much stronger, and it is signalling that there are new technologies and that everything should be about good control systems. So there's some good points.
Yeah, so that's good to hear. So it sounds like we've made improvement. There are still obviously going to be comments. Like, as I mentioned at the onset of our conversation, over the next 3 months, stakeholders will have an opportunity to voice their concerns. During this time period, comments will be collected and then reviewed again. Hopefully there won't be 6,200 of them the way there was a couple of years ago. But obviously there will be items that'll be up for discussion. Where, in your opinion, do you think the largest number of concerns will be focused in terms of the commentary?
Okay, yeah. So perhaps just very briefly say, and with this consultation process, it's slightly different as well. So in 2017, the whole Annex was out for consultation, and theoretically any person in the world could write in. This time round, it's a little bit more narrow in what's out for comment, and it's either invited professional bodies, or some professional bodies have written in and been given approval to comment. So it's slightly narrower, which kind of I think signals that they're closer to issuing the final version. But I think the things that are going to flag for me are the clean room qualification process. What is involved? What does it involve? There are some extra tests there, and are they applicable for all clean rooms? So there's a strong emphasis on doing recovery testing of clean rooms, which is artificially creating particles and seeing how quickly they dissipate. I think there's bound to be comments around the production of water for injection. So there used to be a distinction between US and Europe. So US allowed distillation and reverse osmosis. Europe only allowed distillation, but that's changed in the last few years, and there is now the ability to make WFI with reverse osmosis. But the Annex kind of downplays it and says there are risks, and there's need extra control. So I can imagine some water system manufacturers chipping in on that as well. There's quite a tightening up of things around how vial integrity is assessed. PupSit will definitely be a big hot topic for conversation. Guidance around media fields. And it might also be that with grade A environmental monitoring, there's been a shift. Instead of saying the level is 1 CFU, it now says no growth. And again, that might raise some comments about the accuracy of methods and what does that mean in terms of microbial viability. So to me, they're the ones that stand out, and I'll be very interested to see what kind of comments come in on those subjects.
Right. No, as will I. I think overall, this is a great process for us to be going through as an industry, because it is helping companies focus more on quality, regardless of the actual verbiage at the end. I mean, they're going to have to follow the rules as such when they're implemented. But I think even in the meantime, it's giving pause to the industry, and there's a lot more talk about just in doing whatever you can as a company to increase the quality of the end product. So, and I think that every time we do reviews like this, that does serve as a good reminder.
Yes, I think that's right. Yeah.
So, Tim, and thank you for spending so much time with me today. This is such an important topic to a lot of folks in the industry, and we spend a little extra time on it. And I appreciate you giving us some of that time. In closing, I just want to give you an opportunity to share any last thoughts or concepts or questions that you'd like to leave listeners with today.
Okay. So I think the standout points are, I think that there's a big emphasis on sterility assurance and environmental control, which is really key. There's the emphasis on the contamination control strategy, which I think captures a lot of the things that we've spoken about. But here the Annex is driving for this to be holistic. So in that sense, it's saying what might happen. An adverse trend developing in grade C could impact on grade B unless you start doing something about addressing what's going on in the grade C environment. A lot of controls that we mentioned around people and what people do, how they behave, what they wear, etc. The importance of quality risk management, which is very key, and that needs to start being as a proactive concept, building in quality by design, and linking to the quality system. And I guess overall, as we've touched upon, it's ensuring that quality is at the forefront. So I think they're the essential messages that are coming out from this new text.
Excellent. Well, thank you for that, Tim. Again, not only thank you for your time, I do want to mention to listeners that you have written an article for the upcoming issue of the Journal of GXP Compliance, which will be released later this month in just a week or so. And folks who are interested in following this topic more can certainly read your article and get a lot more information and a lot more perspective on your thoughts in terms of the key elements that are up for change and probably up for comment as well. So thank you for writing that for the IVT network too.
Yeah, you're welcome. Hopefully people find it interesting. There's a lot more detail in the article.
There is. Absolutely. Thank you. So again, thanks for being with us today. I also want to give a big thank you to Ben Kitchen, our producer, and the Studio 21 Podcast Cafe. And a big shout out to all of you, our valued listeners. If you like Voices in Validation, we encourage you to subscribe in your podcast player of choice so you never miss an episode. And don't forget to share it with your colleagues and online networks. For more information on the podcast and links from today's show, please visit www.ivtnetwork.com. We'll be back again next week with more great interviews. Until then, make it a great week.
The views and opinions expressed by the hosts, guests, or callers of this program do not necessarily reflect the opinions of the Studio 21 Podcast Cafe, the United Podcast Network, its partners, or affiliates.